Good afternoon, everyone. My name is Karim Dhanani. I'm from FACTS in South Africa. FACTS is a scientific services company offering services to the food industry. We specialize in food allergen management and analytical services. I'm here to speak to you today about allergen detection by LCMS and its implementation in a routine laboratory. It's not detailed method information, but just a discussion of some of the important decisions we made and the rationale behind them. Okay, so as everyone knows, the mainstay of food allergen detection is ELISA. It's sensitive, precise, and quick, and very easy to use, which makes it ideal for use in the production environment. However, if you're sending samples away to a specialized laboratory for confirmatory analysis or otherwise, you would hope they would use methods that were not prone to the same types of errors, provide results with really high confidence, and ideally actually give some additional information, which might help you diagnose the source of contamination or the nature of contamination. So when we were thinking about implementing LCMS, there were two main objectives. One, we wanted to avoid false negatives and false positives. And secondly, we wanted to reduce the sources of error to make sure that we gave more accurate quantitative information. So what is targeted proteomics? Well, it starts out exactly the same as ELISA with protein extraction, but then the proteins are digested into their constituent peptides. Now, these peptides have a sequence, and the sequence gives them their biochemical properties, which allows them to be separated temporarily using chromatography. The sequence also gives them their mass, and their mass gives them their fragmentation masses, which allows them to be analyzed in a mass analyzer and a mass spectrometer. And these are best in a triple quadrupole mass spectrometer. This is best thought of as like mass filters. So the unique nature of the peptide masses and their eluting times based on their biochemical properties allows for intrinsically high specificity. And this in turn allows for unambiguous detection of those peptides. So the advantages that we wanted to capitalize on related to the direct detection of the peptides are, firstly, of course, you know what you detected, which is often in contrast to immunoassays. Secondly, because there are no antibodies, you can be very thorough with your solubilization procedure. Now, this is important for two reasons. Hopefully, it will reduce variability between matrices, but also it maximizes your chances of detecting something that is present. The solubilization procedure, though, has to be compatible with downstream processes, which is something I'll touch on in a second. The pitfalls we wanted to avoid is it can be difficult to pick appropriate peptide targets. There can be complexity in converting a signal to a result, and these workflows can get expensive. Okay, so what are the sources of error? So in both immunoassays and in LCMS, the choice of calibrant or average of calibrants or whatever the system is using is obviously the first source of error, and that's what can't be corrected for by the method, and that's a separate issue. But then the error is introduced by differences in analyte solubility between the analyte and your calibrant. And then in the actual process or method, most of the error in an ELISA will come from changes in protein structure, denaturation, aggregation, cross-linking, that type of thing. And we don't have that in mass spectrometry. But the other error, there are sort of three areas where we felt were tractable that we could improve on. So one, peptide selection. Obviously, this is an incredibly important step. Two, minimizing error by maximizing analyte solubility. So the buffer system and the process we actually use to make sure we recover peptides. And then three, making sure that we have reproducible sample preparation procedures and controls in place. Okay, so choosing appropriate peptide targets has never been easier. There's lots of work being done, reviews being published, and there are repositories of that information. The best peptide to choose, though, will depend on the protein solubility in your buffer system, and the specific ion transmission efficiency in your system. That's what people refer to as how well the peptides fly. But choosing a specific peptide to use as a qualifier or a quantifier can be a bit of a fraught business, because specificity can be a double-edged sword, and bioinformatic information will never be complete. So people might have seen methods where they're using 1 or 2 peptides to identify the presence of a protein. And although that's not wrong in any way, I do worry about redundancy. So I think more peptides is always better, although it does make it more complicated potentially to convert your signal to a result. And an example of this, although the authors do acknowledge the problem, is in that AOAC official method published last year, where total milk is quantified by 2 peptides, both coming from alpha S1 casein. So obviously we can all see the problem with that, but we want to avoid those sorts of problems. The other reason why that's important is because you do get co-eluting peptides, and you can get isobaric peptides, which causes interference in your signals. So the more peptides you have per allergen, the more chances you have to avoid that interference when it comes to interpreting your results. So we decided to keep a very high level of redundancy in target peptide selection. And this actually meant we threw out maybe one of the biggest advantages of mass spectrometry, which is multiplexing. So we use single allergen detection runs. So here's an example. Our wheat assay on the right-hand side here, it has 14 monitored peptides from 7 different proteins, and that's it. We don't detect anything else in there. So cross-referencing of multiple peptides gives you higher confidence in your results. So this is maybe also the right time to talk about some other comments that we've seen, one of which is the idea that cross-reactivity in ELISA is not a bug, but a feature, and actually only detecting the specific thing legislated, it might be a bad thing. That may be true, and obviously, though, it's fairly simple to add informational peptides to an assay like this. Say if you wanted to tell if the signal was goat's milk or sheep's milk or cow's milk. Okay. So this is the just a depiction of the AOAC reference method. It's quite a common framework. Lots of methods published constellate around this type of procedure. And there are a few things I just want to point out about them, just because we're going to contrast them just now. One, the use of urea is quite common. Clearly, it's not that much of a problem, otherwise people wouldn't be using it, but urea can modify peptides, which can affect their digestion ability, especially in the presence of heat. So if you want to use heat during your extraction, that can be a problem. And also heat is applied during the reduction stage. So there is a risk there of introducing some source of error. Secondly, in terms of handling, it looks like it would be a difficult procedure to automate. But depending on the laboratory's throughput, that may or may not be a problem. And then the last thing is cost. So the size exclusion filters and SPE cleanup filter cartridges that people need to use to clean up their peptides before mass spectrometry can be really expensive, especially if you're processing lots of samples. And then the other thing here is the non-ionic surfactant that is used to aid solubilization in the extraction buffer. That surfactant is really expensive. It's compatible with mass spectrometry because it's non-ionic, but it is, again, if it's going to be applied in a laboratory for real, it can't be that expensive. Okay, so this is our sample preparation procedure. It's based on the central point, the middle part here, which is hydrophobic interaction chromatography, SPE type cleanup. Now, this was a technology developed for proteome-wide unbiased sample prep. It's really well suited to sample preparation for allergen detection. It allows you to bind proteins and wash them to remove matrix contaminants, to remove any reagents you added to the extraction buffer, everything like that. Everything can get washed before digestion, which is key because matrix components, lipids, and all sorts of things can interfere with digestion, and we don't want that. So just to look at the extraction buffer, the differences here are we're using SDS, which is cheap, and we can use it in reasonably high amounts, although we could go higher. We also use, rather than using a chemical chaotrope, we use sonication and heat. The sonication itself is actually enough. We add heat because the only matrix we've observed it improving recoverability in is multiple matrices like chocolate. But it does make a big difference. Out of interest, it was roughly the same difference, heat and no heat, between those two states and between tempered and untempered chocolate. So take that for what it is. Okay. The greatest thing about using magnets is that it's simple to automate. So in our laboratory, we do have, after sample extraction, we spin down any undissolved particulates, and then the samples are put onto a liquid handling platform, and the samples are processed in 96-watt plates before digestion and then recovered, and then they are ready for mass spec analysis. Okay, so the next thing people might have noticed about lots of the LC-MS publications is that the methods are usually optimized for throughput and not confidence. So you know, sometimes, depending on how thorough your cleanup procedure will be, you can get contaminants coming through and messing with your chromatography. And obviously that can have catastrophic effects on your ability to detect or quantify anything. So it's important we use retention time control peptides before and after every run. And we also use an internal test protein that confirms that the reproducibility of the retention time. We also use, it's also very important to confirm that there is no carryover between samples. Now, this is obviously obvious, but in allergen analysis in particular, if you've, say, got wheat and you've got peanut, the matrix in one sample can be the analyte in the next. So we use active QC blanks that are monitoring for carryover. And then the last point that had always bothered me somewhat about the LC-MS procedures was, how do you know if your digestion and cleanup procedure worked in a given sample? Because obviously that's not something that an internal quantification standard or the like would control for. So we wanted something that was cheap and reproducible, that we could always get hold of. And we landed on human insulin, which we add at the extraction stage. And then that comes all the way through, and we get a signal in every run to confirm that the digestion worked in that sample. So the philosophy here is lower throughput, but higher confidence in results. Okay, so calibration, converting your signal to a result. So firstly, we wanted interoperability with previous results from the laboratory for our clients, which meant that we stuck with the same calibrants where possible. And that works pretty well in terms of getting the same sorts of numbers out. In terms of how to actually convert a signal to a result, though, there are lots of options with LC-MS. You can use straightforward calibration curves, but people favor internal quantification standards, which are isotopically labeled peptides that match your target peptides. There are many approaches to convert the result from this to an actual number. That can involve conversion factors from the protein molecular weight, or them converting directly to a calibration curve itself. Given what we're doing, though, with multiple peptide targets and not knowing yet which proteins are likely to be modified in different ways, we didn't want to commit to any one or two peptides for quantification at this stage. So what we decided to do as a compromise was to use the fact that we're monitoring multiple peptides, use traditional calibration curves, and then use internal positive controls that are internal to each batch for IQS-like adjustments. So we add them in there at the limit of detection. And then that gives us another advantage as well, which is that in potentially fractionated allergens like milk or egg, we can see when the proteins that we're monitoring and quantifying don't agree with each other, and we can go with the higher value where we can see that the results don't agree, or the average value when they are closer together. Okay, so a little bit of the method performance. The first thing that has already yielded really great results is the fact that we have this wide range of quantification. We have 1,000 ppm cutoff on the upper range arbitrarily, just to cut down on the number of runs we have to do for our calibration curves. But obviously, with a mass spec, you can go much higher. In terms of actual method limit of detection, that varies by allergen and calibrant, obviously. The peptides that seem to fly best that we've seen so far are the milk ones in our system. We could go very, very low on that, but where we can go lower than 1 ppm, we tend not to, again, just for interoperability and requirements from a practical point of view. During the validation, we did obviously a full method validation and recoveries from multiple matrices. We did incurred samples, proficiency testing samples, and we did do some homemade ingredient-based detections. Some of that is here. The interesting thing on this left-hand panel is we're showing that the extremes of the AOAC matrix triangle don't significantly affect recovery at different spiking levels. On the right-hand side, this is just showing recovery in a baked good. We've included an ELISA on this, not to compare them directly, because there is obviously a dilution factor with the ELISA, so it's an unfair comparison. But the relative change in peptide loss from raw dough to cooked is less. And we think this is due to the more vigorous solubilization procedure. One of the major things we did, that wasn't, we can't draw too many conclusions from it because it's not very systematic, but we did run our pre-existing ELISA methods against the new LCMS method before it was introduced commercially, side by side for a number of months. And the good news here is that the match rate in terms of qualitative results is very high. The vast majority of the difference between results, between the results we obtained, was due to the lower limits of detection on the LCMS methods on the whole. But the first objective of our method development was to ensure that we didn't have any false negatives, and there were a number of occasions where it's a low frequency type of event, but in these difficult matrices, spices and flavorings whatsoever, you do get these nightmare scenarios where you have a lower than limit of detection, lower than range of quantification result on an ELISA. And this is one that stuck out in my memory where there was a very high detection level on the LCMS. But that's uncommon, but obviously 0 is the ideal number of those. And with that, I'd like to thank you for your attention and for the invite, and particularly to the FACTS team for all of their hard work in making this happen. Thank you very much.